Assalamualaikum warahmatullahi wabarakatuh to Dr. Farizan and Dr. Hafzan. I am Sabrina Kamaruzaman from Nutrition Year 4. I'd like to present my research project in nutrition entitled Nutraceutical Potential, Phytochemical Content and Antioxidant Activity in Lettuce, a scoping review. My supervisor is Dr. Marina binti Abdul Manaf. So here is my outline for the whole presentation, which consists of introduction, methods, results, discussion, conclusion, and also references. For the introduction, lettuce, or known as Lactuca sativa, has been cultivated worldwide as one of the main leafy green vegetables. The nutritional values of the lettuce always being underestimated. Even lettuce is one of the most widely consumed vegetables worldwide. A previous study has shown that lettuce can produce anti-inflammatory, cholesterol-lowering, and anti-diabetic activities. In addition to that, nowadays researchers and producers are attracted to enhance the secondary metabolites compounds in lettuce through the genetic modification and novel agronomic techniques. So the objective of this review were to identify the nutraceutical potential in lettuce and to review the phytochemical content and antioxidant activity in lettuce. There were 3 research questions in this review, which were: is there any nutraceutical potential present in lettuce? Which phytochemicals are found in lettuce? And how much is the antioxidant activity in lettuce? Next, we move to methods. This copy review was drafted using the preferred reported items for systematic reviews and meta-analysis extensions for copy reviews checklist. This checklist consists of 20 crucial items followed by 2 optional items in order to complete this copy review. But first of all, the eligibility criteria must be set up, which consists of inclusion criteria and also the exclusion criteria. For the inclusion criteria, any studies related to lettuce as samples were included in this study, while for the exclusion criteria, the study that only described the macro and micro mineral content of lettuce, involved baby lettuce as the sample, and involved complex genetic modification were excluded from this review. Next is information sources. I used the PubMed database searching in May 2021. For the search strategy, I used the keywords that based on the synonyms of this scoping review title. After that, data must be charted into data charting table, and the data items included were author's name, year of publication, objective, study designs, samples used, parameters measured, and also findings. Lastly, the synthesis of result. This part, I divided the table into 2 tables, which are identifying the nutraceutical potential in lettuce and also reviewing the phytochemical content and antioxidant activity in lettuce. So here is the search strategy that I have been used in this review, and this is the Prisma flow diagram that has been created. Next, we move to the result. There is 6 articles identifying the nutraceutical potential in lettuce. So here is the summary of the selected articles. First, Zhou et al. 2019 evaluate the anti-proliferative effects from reduced nitrogen supply and alteration of phenolic compounds lettuce against cacao cells. Next, Cheng et al. 2014 to report on acute lowering and insulin sensitizing activity of generated lettuce cultivars with very high polyphenolic content, named RSL. Next, Ismail et al. 2019 to test the role gene in transformation on the specific bioactivities for type 2 diabetes mellitus treatment. Next, Kin et al. 2018 to investigate the major bioactive metabolites in new dark red lettuce cultivar on anti-cancer effects activity. Next is Hong et al. 2018 to study the effect of ferment lettuce extract, skullcap root extract, and their mixture on sleep behaviors in vertebrate model. And lastly is Kim et al. 2017 to investigate the content of lactosine and lactocopicrin in lettuce on sleep prolonging effects. Next, we move to the second table, which is a summary of selected articles reviewing the phytochemical content and antioxidant activity in lettuce. First, for the Zhou et al. 2019 to investigate the effects of reduced nitrogen supply on the production of phenolic compounds in lettuce. Next, Cheng et al. 2014 to generate the lettuce cultivar with very high polyphenolic content, named RSL, by developing the somatronol variant. And then Kim et al. 2018 to identify the important metabolites and to compare the antioxidant activity of different lettuce cultivar. Next is L. Nachert is to determine the phenolic profiles and antioxidant of green and red Selenova lettuce at two different harvest stages. And then the Cruz et al. 2012 to investigate the effects of soil coffee ground (SCG) on carotenoid and chlorophyll content in lettuce leaves. Next, the Kim et al. 2017 to study the radical scavenging activity in raw lettuce extracts against oxidative stress. Next is Chadwick et al. 2016. This to detect the compounds of interest for the sweetness and bitterness in lettuce. Next, Moreno as Camila et al. 2020 to test the effect of elicitation on polyphenols and carotenoid metabolism in butterhead lettuce. Next, Joko et al. 2020 to study the effects of silver, gold, and platinum nanoparticles on the antioxidant status of oak leaf lettuce plant. Ramos Sotelo et al. 2019 is need to study the performance of the hydroponic lettuce culture with two low salinity shrimp effluents. He et al. 2021 determined the effects of individual or combined UVA and FR radiation on physiochemical properties of hydroponic lettuce. Last but not least is Ayuso Carlos et al. 2020. Study the potential of Rhizobia strain to act as a promoter to enhance the bioactive compounds in lettuce. Lastly, the Seniza et al. 2020 study the changes of phytochemicals in lettuce when react with suboptimal plant nutrient supply. So we move into the discussion part. So first, Zhou et al. 2019. The anti-proliferative effects affected the factors affecting cacao to cells proliferation, such as decreasing the cell viability, anti-apoptotic proteins, mitochondrial membrane potential, and increasing the pro-apoptotic proteins and DNA impairments. The transcriptional levels of two anti-apoptotic proteins, which are BCL2 and BCLXL, while for two pro-apoptotic protein, BAX and BEC, the transcriptional levels were upregulated, which result in higher transcriptional levels and induce the cell cycle arrest. RH123 fluorescent intensity decrease, which indicates the loss of mitochondrial membrane potential and high rates of DNA impairments. For the phytochemical content and antioxidant, the amount of phenolic accumulation in lettuce, such as chicory acid, chlorogenic acid, quercetin, and luteolin, was measured in this study using the HPLC analysis. According to the analysis, chicory acid, chlorogenic acid, quercetin, and luteolin were the most abundant phenolic compound found after 5 days of receiving low nitrogen treatment. Next is Cheng et al. 2014. This is due to the chlorogenic acid contained in RSL contribute to the anti-diabetic effects in the mice. This effect can be seen through the inhibition of hepatic glucose production, reduced early glucose response, and plasma bioavailability. In addition, caffeoylquinic acid, flavanol, and anthocyanin display their roles to prevent the breakdown of starch and disaccharides through the inhibition of alpha amylase and alpha glucosidase, and sugar absorption by transporters, sodium glucose co-transporter 1 and glucose transporter 2. Thus, an increase in fasting blood glucose and postprandial blood glucose can be avoided from the accumulated activities of polyphenols in the intestinal lumen. For the second part, which is phytochemical and antioxidant activity content, the quantification of main classes of polyphenols compounds like hydrocinamic acid, flavanols, and anthocyanins was measured. Next is Kim et al. 2018. This assay measured the cancer cell viability such as human lung adenocarcinoma cell line, human hepatoma cell line, human cancer colorectal adenoma cell line, and colon cancer cell line. This indicates that as the concentration of B2 extract increased, the viability of the 4 tumor cell lines also decreased. This can be seen when tested with MTT assay. The mitochondrial succinate dehydrogenase converted the insoluble violet crystalline formazine produced from MTT and deposited in the living cell, but not in the dead cell. And another one is triple blue assay. This blue dye penetrated into the non-viable cell due to loss of membrane integrity. And then for the B2 and B3 lettuce contain the strongest antioxidant activity when compared with other cultivars, and the S2 lettuce cultivar was the lowest in the antioxidant activity. Other than that, B2 and B3 lettuce cultivar were also rich in anthocyanin and mostly in cyanidin derivatives. Next, Kim et al. 2017, the seed and leaf extract from normal lettuce were used and showed an increase in the sleep duration at low and high concentration respectively. Lactocopicry resulted in no analgesic and sedative effect, while for lactocin, it became the predominant substance to display the sedative property, including sleep-promoting effect. This study reported that both low and high doses of both extract derived from normal lettuce can produce a significant reduction in sleep latency and increase the sleep duration. And then, in the other hand, the leaf extract possesses a higher level of flavonoids than the seed extract, but the seed extract contains higher polyphenol content. The seed extract can be reported to have a better antioxidant activity than leaf extract because polyphenols, flavonols, and anthocyanins were defined as compounds that had higher antioxidant activity. Next, Hong et al. 2018. This study combined the romaine lettuce leaves and skullcap root in the forms of extract in order to extend the potential of efficacy. Based on Chen and colleagues 2016, the skullcap root is known as traditional Chinese medicine and be a remedy for diarrhea, insomnia, dysentery, high blood pressure, and hemorrhaging, respiratory infections, and also inflammation. RE and SE mixture show more effectiveness as it increased the binding ability to GABA receptor. This is because a single mixture resulted in the lower displacement of 3H-fumazanium, which is a radioligand for GABA receptor, when compared to RE and SE mixture at all tested concentration. Next is El Nacher et al. 2020. The carotenoid content, total ascorbic acid content, and phenolic compound in green and red microgreens and mature leaves were measured. The mature red leaf lettuce were higher in lutein and keratin compared to green leaf lettuce was due to carotenoids are naturally abundant in red pigmented leaf, and carotenoid concentration can elevate 3 to 4 times upon maturation. Ascorbic acid was produced and built up at the beginning of red microgreens and continuously increased in the red mature leaf. However, for the green lettuce, total ascorbic acid remained constant because phenols acted as scavengers of reactive oxygen species to protect the leaf from light damage. So, ascorbic acid in green Selenova reduced from microgreens to mature stage and remained constant at the end. And then Ismail et al. 2019. This study tested the specific bioactivities, or known as anti-diabetic targets, such as inhibition of alpha-glucosidase, DPP4, and stimulation of glucagon-like peptide 1 secretion in streptozotocin-induced diabetic mice. These compounds were elevated after the raw gene manipulation took place. Next is Cruz et al. 2012. Lutein concentration range in SCG treatment was significantly increased than control. Similarly, the beta-carotene concentration range in SCG treatment also was significantly increased than control. Next, Juko et al. 2020. Silver, gold, and platinum nanoparticles were used in the study. These nanoparticles act by interfering with the electron transport chain in the mitochondria and chloroplasts that resulted in an oxidative burst, which caused a rapid release of ROS into the cell compartment. So for overall, oat leaf lettuce extract that treated with these nanoparticles produced a higher scavenging of the PPH radicals by 37.5% and 44% respectively. Next, Moreno S. Canilla et al. 2020. This study used elicitor as a use stressor, which can increase the time improvement of functional quality due to the activation and defense responses. This concentration give insight on phytochemical content like terpenoid content, flavonoid content, and carotenoid content, and also polyphenol family like flavonoids, hydroxybenzoic acid, and hydroxycinnamic acid using HPLC analysis. So for overall, MJ, which is the methyl jasmonate, was the elicitor that produced the greatest effect at all concentration on phytochemical and phenolic family in green and romaine lettuce. Next is Chadwick et al. 2016. This study detected the compounds of interest for sweetness and bitterness in lettuce. So the study assessed the glucose content and cis-quaterpenoid lactone, SL, using HPLC analysis. SL was the compound that contributed to bitter taste, while glucose content contributed to sweet taste in lettuce. In addition, SL produced by lettuce can act as antiphedants and phytoalexin, which are known as antimicrobial, and take part in the defense system. Next, Ramos Sotelo et al. 2019. This study studied the performance of hydroponic lettuce culture with two low salinity stream effluents, which were stream effluents from well water and also diluted seawater. Overall, well water stream effluents were reported to have a higher level in total phenolic content, total flavonoid content, and antioxidant because well water effluents consisted of higher concentration of sodium ion and chloride ion, which affected the nutrient content and also plant growth. Then, he et al. 2021 determined the effects of individual or combined UVA and FR radiation on physiochemical properties of hydroponic lettuce, which are yanzi and red butter. So for overall, UVA treatment had interactive effects on phytochemical profile in lettuce and can be used in an artificial lifetime factory to increase the functional crop. Last but not least is Ayuso Colors et al. 2020. This study make a comparison of phenolic acid and flavonoid content of Rhizobia strain under normal and 100 millimole sodium chloride condition. Phenolic acid and flavonoid content were significantly increased in lettuce inoculated with Rhizobia strain under normal condition compared to non-inoculated and under 100 millimole sodium chloride treatment. Some phenolic acid like caffeoyl malic acid, caffeoyl tartaric acid, and coumaric acid, protocaticoic acid glucoside, and epigenetic derivative show an increase by 180% in lettuce inoculated with Rhizobia strain under 100 millimole sodium chloride treatment compared to under normal condition. This is because under salinity treatment, the ROS scavenging mechanism in lettuce has been activated to overcome the stress. So the phenolic acid increase. And then lastly is Saniza et al. 2020. This study involved two genotypes which are green and red selenova and treated with three different strengths of nutrient solution which are full strength, half strength, and also quarter strength. So for overall, the nutrient strength solution treatment only work for several phytochemicals, and red selenova show the most responsiveness to the nutritional deprivation compared to green selenova. So next, we move to limitation. In this review, there were three limitations, which were this review only consisted of one reviewer with the supervisor's guidance. Next, this review only used one electronic database searching, which is the PubMed database, during selecting the sources of evidence in order to prevent the massive duplication of articles. And then, lastly, the search strategy of this review was not sensitive enough to identify the full breadth of research activity. As conclusion, there were several nutraceutical potentials found in lettuce throughout this review, such as anti-proliferative, anti-diabetic, anti-cancer, and sleep-promoting effects. In general, these effects were derived from the presence of phytochemical and antioxidant content in lettuce itself. For phytochemical content, phenolic compounds were the most finding in lettuce using the HPLC analysis, while for antioxidant activity, anthocyanins and flavonoids were the major findings by using the DDP-PH and ABTS assay. However, evidence was insufficient to assess for the phytochemical and antioxidant content in lettuce. This is because most of evidences obtained were from different genetic modification and novel agronomic techniques. So here is the references that have been used in this presentation. With that, thank you and have a nice day.